Thank you, Emily. And thanks everybody, like Emily said, for showing up tonight. Huge thanks to the folks at Hawk Ridge for inviting me to come and talk with everybody. Um, so for those of you that I haven't met, which is probably most of you, my like Emily said, my name is Kara Snow, and I work with the Avian Ecology Lab at NRRI here in Duluth, Minnesota. And tonight I'm going to talk about, I feel like recapping, but about two projects in our lab. One using environmental DNA or eDNA to help detect invasive species in Minnesota, and another project that's focused on pileated woodpeckers. And they're pretty different projects, but both have actually benefited from collaboration, including support from Hawk Ridge, which is something that I will touch on as I go. All right, so let's start with the basics. I'm sure lots of us know this, but what are terrestrial invasive species and why do we spend so much time and effort trying to find them? So, at the most basic level, these are species that are introduced outside of their native range, and once they're here, they cause ecological, economic, and even sometimes human health impacts. And one of the biggest challenges with terrestrial invasive species is that once they're established, they're pretty hard to control. And they often spread really quickly because they don't have these like natural predators keeping them in check. And by the time that we notice them, they've already gained like a foothold. So in Minnesota specifically, terrestrial invasive species are a huge issue. In this part of, I was going to ask if anybody knew how much money we spend on terrestrial invasive species in Minnesota, but everybody is muted, so I will just give the answer away now. And then I guess later people can tell me if they got it right or not. Um, so terrestrial invasive species in Minnesota actually costs the state about $3 billion every year. So like when you add up the impacts to forest, agriculture, and all these management costs, it's super expensive. And some of the big ones that you may already know about include the emerald ash borer, the spongy moth, which was formerly known as the gypsy moth, and the brown marmorated stink bug. These species don't just affect the ecosystems, they affect homeowners, land managers, and also entire industries. So this brings me to the Minnesota Invasive Terrestrial Plants and Pests Center, or MITPPC. So if you thought that NRRI was a difficult one, this one will give you a run for your money. So MIT PPC was founded by the Minnesota legislature in 2014 with a very specific mission, which is to research the prevention, detection, and control of terrestrial invasive species in Minnesota. And what's really unique about this center is that it's the only research center of its kind in the country that's focused specifically on terrestrial invasive species. Funding for this comes largely from the Environmental and Natural Resources Trust Fund, as recommended by the LCCMR. And the center supports collaborative applied research, meaning projects that can actually inform management and decision making on the ground. And our eDNA project is one of those efforts. So, how do we usually detect invasive species? Traditionally, invasive species are detected using things like traps, visual surveys, citizen science. So for example, the Minnesota Department of Agriculture deploys about 20,000 spongy moth traps across the state each year. And these traps are designed around the moth's biology. And so they use these sexy synthetic pheromones to attract male moths. And these methods are incredibly important and effective, but they do have limitations. So they can often only target a specific life stage of of an invasive species, like for example, with the spongy moths, the adult males. And they can also miss low density populations or early invasions, and they re also require like a lot of time and labor and taxonomic expertise. So this raises an important question. What if there was another way to detect invasive species, one that could complement these existing tools and help us find species earlier or in places that we might already miss? And this is where eDNA comes in. So when we talk about eDNA, we're talking about environmental DNA, which is genetic material that is found in environmental samples. So that can be from things like soil or water, or in our case, we're focusing a lot on poop. Why poop? Well, when an animal eats, the DNA from the organisms that they ate passes through their digestive system and then it comes out the other end. So bird poop, bat poop, any poop actually will contain the DNA from the food that they ate. So insects, plants, chicken dinner, whatever. And on top of that, organisms are constantly shedding their own DNA as well through like skin cells, hair, saliva, other biological materials. So these samples end up acting kind of like a biological snapshot, telling us who was in the environment slash what was being eaten, even when we can't even see the organism itself. So we're actually learning about invasive species from poop in this project. So with eDNA, there are kind of like two general approaches. So one is very targeted, and you're asking a specific question like, is this species here or not? And then the other approach is like a little bit broader. So you collect a sample and then you use the DNA in it to screen for many species within a particular taxonomic group. So like insects or fish or fungus all at the same time. And so that's the approach that we're using for this project. And from a single sample, like a single bird poop, you can screen for loads of potential invasive species within a group, which is especially useful for early detection and for species that are hard to find using those traditional methods that I mentioned earlier. All right, so at a high level, the process looks kind of like this. So you collect the environmental samples, in this case poop, um, extract the DNA from it, amplify a target region using PCR, sequence that data, and then compare those little sequences of DNA to reference databases to identify which species are present. So from a single sample, you can, like I mentioned earlier, detect dozens and sometimes hundreds of species. For this project, we are using a variety of sample types and collection approaches. So for birds, we collect fecal samples through mist netting efforts, through collaborations with maps and other banding stations, and also we've been collecting poop samples opportunistically during our other projects. For the bat portion, which I'm not involved with because I am a bird person, those folks are collecting their poop pellets beneath the bridges that they roost in or like spend the night in. And we are collecting samples from a mix of priority locations across the state. Some of those are places where we already know invasive species are present. So, like, for example, forests where there are documented emerald ash borer outbreaks. And then sampling in those areas is going to help us ground truth the method and confirm that, by using bird poop, you can actually detect invasive species that we know are there. And then we're also sampling in areas where outbreaks haven't been documented yet, but where the conditions suggest that they could occur. And that gives us a way to like look for early warning signals. So species that might be present at those low levels before they're detected through those traditional surveying methods. All right, so this approach has a lot of promise, but it's not perfect. So I wanted to talk briefly about what eDNA can do really well, but also where we kind of need to be careful and its limitations. So because, especially for this project, we're using a variety of different sample types. We can survey across multiple life stages. And from a single sample, like I mentioned before, you can detect many species at once. And this is especially useful for rare or elusive species. And it's also relatively quick and it is minimally invasive and it doesn't require you to like capture species themselves, like the invasive species themselves. But there are also some limitations. So eDNA breaks down over time. And timing really matters. So if an animal eats an invasive species, there's only like a window of time when that DNA will pass through the digestive system and come out as feces. So if you miss that window, you could miss that signal. And it's also difficult to estimate abundance using eDNA. So you can only see like was the species in the poop or was it not? You can't say, oh, they ate a bunch of it or there's a lot of it here. It's mostly used for presence absence. And you can't always pinpoint exactly where the detected species came from. So, we're sampling from flying creatures, birds and bats. So while we might be able to say, oh, this species is in their poop, we can't exactly know in some cases where that species was consumed. So keeping those benefits and limitations in mind, this is what our first year of data actually looks like. So in our first year, we have processed almost 700 samples, including bird poop, bat poop, and we also have some small mammal stomach contents, and have also sampled from insect traps. And so far, we have completed taxonomic identification for over 400 of those samples, and we have detected 3 priority invasive species. So the spotted winged Drosophila, or fruit fly, the spongy moth, and also as a bonus from supplemental data from a different eDNA study we've done, we have detected the brown marmorated stink bug. So taken together, these early results are really encouraging. They suggest that eDNA from wildlife samples can be useful for detecting terrestrial invasive species, particularly as a complement to existing monitoring efforts. So, looking ahead to the next field eDNA field season, time is going to be really important. So, for example, woodpeckers feed like most heavily on emerald ash borer larvae in the late winter and early spring when those larvae are larger. So we're going to be focusing our targeting sampling during that window to hopefully improve detection of EAB. And we're also going to be expanding into sequencing for invasive plants, which I think is really exciting because I have not done eDNA work with invasive plants yet. So beyond invasive species, we have used eDNA for a number of other projects in our lab. So including looking at tree swallow diet across land use gradients, looking at boreal chickadee nestling diet under drought and non-drought conditions, and also linking common tern diet to mercury exposure. So eDNA has really become a versatile tool in our lab. All right. That's kind of like the eDNA portion. And now I want to switch gears a little bit and talk about a very different project from our lab, which is our work on pileated woodpeckers. I will start with a little background on why pileated woodpeckers are so important. So these woodpeckers are considered a keystone species in Minnesota's forests, and that's because the cavities that they create for nesting and foraging are used by a huge range of other wildlife, from bird species to mammals. And most of these other species that use their cavities can't make their own cavities, so they rely so heavily on these cavities made by pileated woodpeckers. So the goal of this project is to assess pileated woodpecker habitat across a gradient of forest management. So a heavily managed to a less managed forests, and use those findings to inform management recommendations that support cavity nesting wildlife. So this is just the first year of the study, and we've been pretty busy. We've been putting in a lot of effort to trap and tag adults, a lot of effort, and not too many birds caught yet. It is actually kind of tricky to catch a pileated woodpecker, it turns out. We have been locating their nesting cavities, documenting their habitat use and their home ranges, and surveying for woodpecker density across our study areas. And we have also deployed trail cameras at the cavities, the nesting cavities that we found in the last breeding season, the first year of our study, to document the use of those cavities by secondary cavity nesters. I have a little video to show everybody. One um, secondary cavity nester that I have shown in this video is a flying squirrel that surprised my coworkers while they were doing cavity measurements and putting the trail cameras up in the early winter. So hopefully this works. It's always like, is it gonna work? Let's see. Oh, here we go. Oh, hi. Yeah. I'm gonna try playing it again if I can. I don't know if I can. Oh well. Um, it's kind of like choppy in that. A little less exciting than a flying squirrel, but we've also been characterizing the nesting cavities that we have been finding by documenting like the tree species and the diameter of the tree that they're nesting in and the height of the tree and the decay stage. And I also just for everybody's enjoyment, and hopefully these videos are a little bit better, but I wanted to share some little video clips that I took last field season of some pileated woodpecker nestlings fledging the nest. I um, was just super fortunate to just like be checking on this nest that I had found earlier on the exact day, the exact moment. It was probably a 10-minute window there when they were actually leaving the nest. So this is like their first flight ever. Um, and I don't know if anybody has ever had a chance to watch a nest fledge, but it's actually a really like quite moving to be able to witness an event like that. So hope my little cell phone video might not do it justice, but hopefully you can hear it. You can hear the parents calling out, "Come on out." Oh, oh no. I don't know why it's, wait, let me try it again. Oh, well, if anybody, let me try this one. If anybody wants, can you see it all right? Is it working on your end?
Oh, we can see the little head poking out, but
Okay.
if you do want to share these with me
Oh,
later,
absolutely.
I'm happy to share them with everyone so that they can see
Okay.
them.
I wonder why it's slower when you're sharing.
Oh no,
I
their
feel
little
like
heads peeking out is very
I
cute.
know. Oh, wait, maybe this one will work. I feel like it will just be like a little smear. Oh,
Oh,
um, you guys just kind of see it, but yeah, I would be happy to share these with anybody who's interested in seeing that. Um, but anyways, okay, so uh transition. The nesting part tells us like a little bit of the story. But what another thing that we're really interested in comes from the adults and learning more about their movement and their space use in different kinds of areas. So whether that's, you know, really pristine old growth forests or even like around the city of Duluth. Oh, right. There we go. So this was the first pileated woodpecker that was caught for this project, and she was caught on St. Patrick's Day last year. And here is a little snapshot of some of the GPS data that we have collected from her so far. And the data that we are going to be collecting from tagged woodpeckers is going to help us to better understand, like I mentioned before, their space use and their movement. And oh, I also wanted to take a moment to acknowledge the help that we've had from the folks at Hawk Ridge. So their team has assisted in capturing pileated woodpeckers for tag deployment, which has been incredibly helpful as we have kind of gotten this project off the ground. As I mentioned before, it turns out compared to other birds, um, getting pileated woodpeckers into the net is kind of a challenge. So support from Hawk Ridge and just collaboration in general, sharing expertise, time, local knowledge has really made a difference for us in this project. So these are two pileateds that were caught last fall at Hawk Ridge. And also for the last week, we have had an active setup in the research area of Hawk Ridge. Um, like I said, this is a learning process for us. There isn't a one way to catch a pileated, even if you go through all of the research. It turns out to be a very challenging bird to help to lure into the net, but we're trying everything. We're using suet carcasses, we're using call playbacks. At Hawk Ridge, we've had some really close calls. There is a pair there that are sometimes interested, sometimes not. I was out there yesterday, I was out there earlier today. We're really hopeful that we'll be able to catch that pair that's been kind of checking out the nets. Um, but yeah, stay tuned on that. Wish us luck. If anybody has any um, secret or tips on how to get a pileated to come down into a net, I would be very interested in that. Uh, and with that, to leave a little bit of time for questions, I just wanted to say thanks to everybody for um, skipping. I don't know what's going on tonight. Curling? No, nobody would. No Minnesotan would do that. Um, but the tuning in for the this talk, and thanks to Hawk Ridge for their collaboration with help with sample collection and help with those pileated woodpeckers. And I'm happy to answer any questions or get woodpecker trapping advice.
Thank you, Kara. Yeah, everyone is welcome to either put some questions down in the chat box or unmute and ask Kara any questions or give any pileated woodpecker trapping advice that you might have for her. Um, I can start with a question I had, if I can remember what it was. Oh, I was curious about, so you said that for the invasive species work, um, when you're using traps to kind of assess invasive species presence, um, that is targeting a specific life stage. And I'm assuming that that's targeting more of like an adult life stage versus what eDNA would be targeting. So I don't know if this is a question. Maybe this is just me talking through it, but it seems like those two things are a really good complement for each other, if they're looking at those different life stages, and could be used in the same place. Is that accurate?
Yeah, totally. So like using like the spongy moth trap, like that those those kind of like triangle traps that you see around are targeting the flying stage, right? Um, and if you think about like in general, what most like songbirds are um, eating, it's going to be the larval or like the caterpillar stage of that. So that's complementary to that. But then also when we use um, uh, poop samples from birds and bats, so you know, primarily a lot of the birds that we're sampling are, gleaning off of bark um and leaves and stuff. So taking like the more sessile or like the larval stage, but then we're also using bat poop and bats are, foraging on the wing, so they're also getting the adult stage of that. So yeah, it's complementary to those traps, which are really focused, but then also when we use different animals feces, then we can also like those different poop can also complement each other with getting like the kind of like full picture, which is pretty cool.
Awesome. David has a question for you. How successful has the bat sampling been for getting good samples of DNA?
Like in terms of like how many sequences there are. The bat sampling has been has been very good. We've gotten a lot of reads from bat samples. The process so these are like the mammal folks, and this is their sampling technique. They've been going underneath the bridges and collecting multiple different bat pellets in the same tube and then homogenizing it and then we extract DNA from that. So there are multiple individuals' feces in that we're taking the samples out of. And it's been very, very successful. As you can imagine, we get so many species detected in those homogenized samples. So it's been really good. Like I mentioned earlier, eDNA degrades, but like not so quickly that some poop that's sitting underneath a bridge for a day won't have DNA in it. I don't know. Hopefully that answers the question.
Yeah. Well, if it didn't answer your question, David, just speak up. Um, Allie says, how are you deciding where in town to trap woodpeckers? I have one in my neighborhood if you need to increase your sample size.
We're there. Oh my gosh. The answer is yes, we do. Especially if they have been coming to your feeder. If they're habitualized to coming to your feeder, we think that that might be an important key, like a, yeah, important piece of that. So, right now we're just trying to anybody who's done tagging efforts knows how much a tag costs and trying to get it out. Like we're just trying to get our tags out on woodpeckers. So um reach out and we will come.
That's good to know. If we have any on some, yeah, land that is accessible, we'll let you know. I know at
Definitely.
this time of year, I definitely feel like I'm seeing them everywhere, but they're maybe not in the best place for trapping. Um, I had another question, or maybe it's not entirely a question again, but I was curious about the invasive species sampling for plants that you're planning to do. Um, and just wondering if you talk maybe a little bit more about that and what species you're hoping to target, if any, or again, it's just kind of a survey of what might be out there.
Yeah, I think okay, so in general it is just like a survey of what might be out there. And for the most part, this study is just like, is this a is this a good tool? Can we do it? Can we like prove that we can detect invasive species using animal poop? I think a big one will probably be buckthorn. That
Yeah.
will kind of be like the ground truth thing, which I was talking about earlier, because we see animals eat their berries and stuff. Um, and so just kind of like a general what is out there. We're, you know, certainly will be excited to find any of those plants on that priority list that the MIT PPC
Mhm.
has. So,
Yeah. At Hawk Ridge, when we're doing maps banding, we have still a lot of buckthorn in the area, and sometimes we, yeah, find um, seeds from buckthorn in the poop. So,
oh,
yeah, it
absolutely.
seems like you're definitely from some, thrushes, you're gonna find some DNA from
Oh
buckthorn
my
in there for
God.
sure.
Fresh
Uh,
poop. Totally.
yeah,
I mean, like you can just like look at it and you're like, oh, you were eating buckthorn.
yeah.
In one of our birds that we caught, it was a gray catbird, and it must have like just eaten a buckthorn berry because it like spit it up in the bag. So it's wild.
Uh, or does anyone else have any questions for Kara? I was kind of curious about the nesting cavities. It looked like that first video that you tried to show us was really high, the one that had the squirrel in it.
Yeah.
Um, have you noticed any trends in cavity height?
No trends yet, um, other than, well, so this is, you know, definitely just like anecdotal, that it does seem like when they choose, and this just like makes sense because it's okay, let start over. When they're choosing to nest in like a more decayed tree, it's usually smaller, like a shorter snag, as opposed to like an aspen that's still partially alive, but in like an early decay stage. And those aspens that are in like the earlier decay stage they do seem to choose to start their cavity up really high, like I don't know, 13 meters plus.
Wow.
So yeah. Yeah, we weren't expecting that. I was going to put in some videos of us try like of our like successful attempts to put trail cameras up on cavities that high, but it was a little bit more complicated and clown-like than we had expected.
Yeah, it looked really high up there. Well, if no one else has questions for Kara, I'm sure that she would be happy to answer any questions that you have later. She's got her email address up here if you think of anything that you were wanting to ask. But yeah, thank you so much, Kara, for sharing about those two really cool projects. I was just gonna quickly put up a slide. Hopefully,
Oh, do you want me to stop? Fine.
um, I should be able to share, maybe.
I'll do a stop share.
Just about the upcoming presentation. So our last presentation will be in 2 weeks from today, February 24th, also a Tuesday at 6 p.m. This is going to be me talking about one of our Hawk Ridge projects, linking breeding and migratory bird populations in Minnesota. And I'm mostly going to be focusing on some of the transmitter work that we've been doing with 3 species of raptors. And then I'm going to end kind of with get excited for the spring migration count, which is coming up in March. So it'll be here before we know it. And let's all get excited about it. And then I also wanted to mention our last speaker was talking about our Broadwing Hawk project that we have coming up this summer. We launched our t-shirt campaign, which is a beautiful t-shirt design by grad student Abby Odegaard. And yeah, we sold a lot of shirts already, which is really great. But if you want to be a part of this and grab your t-shirt, there's always still time. So the t-shirt fundraiser will stay up for the duration of the project. It's currently shipping in batches. So yeah, get your limited edition t-shirt now. Um, Yep, so with that, I hope everyone has a lovely evening and hopefully see you all in 2 weeks for our last presentation. So thank you all and have a great night.